Well, good morning. I'd like to welcome you. I'm Bill Brody, president of the Salk Institute for Biological Sciences. And we're here really to talk about a really exciting discovery that's being released today in two major papers by Salk scientists. A number of people always ask me, what does the Salk do? And I say, well, the Salk is about discovering at the most basic level how cells in the body work, whether it's molecules, whether it's the nucleus, whether it's DNA, whether it's proteins, how cells function. And people say, well, what's the relevance to that? And I say, this is the upstream discoveries that ultimately lead to improvements in human health. And I'm reminded in the 90s, one of our scientists, Tony Hunter, was studying cancer and discovered that there are a series of altered enzymes called tyrosine kinases, which occur in certain forms of cancer. And today there are about 200 drugs in either clinical trials or approved for use against cancer based on these discoveries that came out of Dr. Hunter's lab many years before. And today, I think through the discussion with our scientists, you'll hear about some exciting developments in diabetes that we hope ultimately will lead to improved methods of treatment of patients with diabetes. Before I introduce our first speaker, I would also like to just acknowledge the support of the Helmsley Charitable Trust, which has been so important in this work. Although the majority of funding for the Salk Institute comes from the government, mostly in the National Institutes of Health, as well as support from the state of California and the Stem Cell Initiative. We depend on private philanthropy as well, particularly for very cutting-edge, high-risk kinds of experiments that require initial data before they can get subsequent government funding. On my right, who I'll introduce now, is Professor Marc Montminy, who's a professor in the Clayton Foundation Laboratories of Peptide Biology. He's a member of the National Academy. He's one of our most distinguished scientists, and throughout his career has been interested in the complex network of how brain signals, hormones, and physiologic mechanisms all work to control the metabolism and energy balance of the body. And this, of course, plays a major role both in the normal function of cells, but also in diseases like diabetes. So with that, let me introduce Professor Montminy.
Thanks, Bill. I'd like to start by thanking Dr. Biao Wang, who's the postdoctoral fellow in our lab that did most of the studies for this paper, as well as our major collaborator and Drosophila expert, John Thomas, who's a professor here at the Salk Institute. About up to 30% of adults in the United States suffer from obesity, which is really a major risk for the development of type 2 diabetes. About 25 million individuals suffer from type 2 diabetes, and more than 1 in every 10 U.S. healthcare dollars is currently spent towards the treatment of diabetes complications. So identifying new genes that contribute to this disorder will help in the diagnosis and eventually therapy for this important illness. We use the fruit fly Drosophila melanogaster to study how fat is stored and burnt because flies actually share much of the same metabolic as humans do. They have many of the same genes. And moreover, their short lifespan makes them an ideal source for using genetics to identify new genes that contribute to obesity and diabetes. So both flies and humans have metabolic systems that resemble a hybrid car. During the day when we eat, we use high-octane glucose to drive the metabolic engine. And at night when we fast, we switch to the battery, which in this case is stored fat. The fat provides a slow but reliable source of fuel for the body. And in response to fasting hormones, a group of fat-busting enzymes called lipases ensure a supply of energy from the battery to the other organs in the body. These lipases are turned on by a genetic switch called FOXO, which is essentially the central transmission of the metabolic system. Now, when the flies eat food, an enzyme called SIC3 shuts off the FOXO switch, and in the process, it cuts off the energy supply from the battery. And As such, by cutting off the energy supply and silencing those fat-busting lipases, it allows the battery to recharge. So we found that flies that lack the SIC3 enzyme can't store fat properly because they're always burning it. And as a result, they die quickly when food is withheld. Now, perhaps the greatest surprise for us is that the SIC3 enzyme doesn't shut off or control the FOXO switch directly. Rather, much like a runner in a relay race, it passes the baton on to yet another enzyme called histone deacetylase 4, or HDAC4. And this enzyme, in turn, controls the FOXO switch by causing a chemical change known as deacetylation. So this is a very complex molecular machine, much like an onboard computer in that hybrid car that has to decide the relative balance of gas and electricity you use. This one is so complex, we think, because it has to govern the switch from burning the high-octane glucose to switching to the battery. I think perhaps the best information that a group of molecules, like this molecular machine that we're looking at, is important is that you see it over and over again in different organisms. And so we found that this HDAC4 SIC3 FOXO machine is also present in mice, and that it's important in driving the metabolic engine. And so disrupting any one of those molecules in the liver of mice was sufficient to disrupt that switch from fasting to feeding metabolism and back. So what does this have to do with the therapy against diabetes? Well, first of all, we can look at SIC3 and the HDAC4 enzymes, and we can see whether mutations in those genes are predictive for the development of obesity or type 2 diabetes in humans. And if so, these would be useful as diagnostic tools for individuals that are susceptible to this disorder. And because they're enzymes, SIC3 and HDAC4 make excellent drug targets. In fact, the HDAC4 enzyme itself has been used, and its relatives have been used as targets for drugs that are currently being used in clinical studies for treatment of cancer as well as certain immune disorders. And as you'll hear from Reuben Shaw, whose studies you'll hear next, these things look very promising for the eventual treatment of individuals with this disease. So I'll stop there and introduce Reuben Shaw, who's an assistant professor in the Department of Molecular and Cellular Biology at the Salk Institute. He's also a Howard Hughes investigator and a member of the Helmsley Charitable Trust fund that is supported here at the Salk Institute. Reuben?
Thank you, Marc. So the major focus of my laboratory is to understand how a critical switch in all the cells of our body governs how our cells, when they grow, and it's a sensor of the cell's energy state. And it turns out that this switch, which is an enzyme called Ampk, this switch is activated by the most widely prescribed type 2 diabetes drug in the world, both in the States and the world. That drug is called metformin, and it's actually an age-old drug that's actually been around as an herbal remedy even since the Middle Ages. It was known to lower blood glucose, actually sweet-smelling sugar in the urine originally was how it was discovered. So it's derived from a plant found in Western Europe called the French lilac, and medicinal chemistry really about 100 years ago distilled down to this compound metformin, which has been used for several decades now, kind of as the frontline treatment for type 2 diabetes. Now, because this is an age-old drug, it was not designed by scientists as a rational therapeutic for diabetes. People had no idea how this drug worked. They just knew that it worked to lower blood glucose. Now, with modern biomedicine tools, we have been trying to discover how exactly this drug is working. And the one thing that we know that the drug does is that it turns on this enzyme switch, Ampk. So we wanted to know how Ampk is then in turn controlling blood glucose. So we searched for new things that Ampk was regulating in the liver to control blood glucose. And that led us to the finding that Ampk was directly regulating the same HDAC enzymes that Marc just told you about. And Ampk was inhibiting them. So this means that one of the ways that this age-old diabetes drug works is to inhibit the activity of these enzymes. And then we dug further into how that was working, and it turns out that they are directly controlling the ability of the liver to synthesize its own glucose. So the liver is actually a very specialized organ in our body that has the ability to make glucose from scratch. So most of the cells of your body actually don't have that ability, and so it's in fact the liver, while we're sleeping, that actually makes glucose from scratch that keeps the brain alive. The brain and most of the cells of our body need glucose to survive, and when we're not actually eating food, our bodies need to make it themselves. That process is mediated in the liver. And it turns out that these HDAC enzymes control that process. So that implies that what we've discovered here is that one of the mechanisms of this age-old diabetes drug is actually to block these enzymes in the liver that make glucose. And that makes sense because the effect of the diabetes drug is to lower blood glucose. So this is one of the ways that the diabetes drug lowers it. It actually blocks enzymes in the liver that help make the glucose in the first place. So finally, we wanted to test whether or not, instead of treating with the age-old diabetes drug, the reason we were doing this was to try to identify new potentials for diabetes drugs. So the question is, if we only block this one step, these enzymes, these HDAC enzymes, would that by itself be enough to lower blood glucose? Or is it that you need to block a number of different things, all regulated by metformin? So we asked in isolation in 4 different genetic mouse models of diabetes, can we lower blood glucose by inhibiting these HDACs? And incredibly, blood glucose levels were dramatically dropped, almost back to normal. So it was more than cut in half, and that is a really striking finding. There are very few genes or enzymes that control blood glucose anywhere as close to that level. The most exciting part of that finding is that, as Marc was alluding to, these same enzymes have been studied for other reasons by oncologists and clinical researchers in cancer trying to find new cancer drugs. And it's found that some of these HDAC enzymes regulate certain forms of leukemia and other forms of cancer. And so pharmaceutical companies have actually been ramping up their efforts to find new versions of these HDAC drugs. Our results predict then that some of those drugs, probably not the same ones that will work on cancer, but some of the ones that are sitting on the shelf that maybe weren't effective for cancer, but in fact hit these enzymes, that they could be potential therapeutics for diabetes. And that means that the time from this initial discovery until the time that this could be tested in the clinic is much shorter. So with that, I just want to say this work was pioneered in my lab by a single senior graduate student, Maria Mihailova, and we could not have done this without this unique collaborative effort between Marc Montmany's lab and John Thomas's lab, as well as Ron Evans, who's sitting to my left, who I'll now introduce. Ron's lab has really been pioneering breakthroughs in the area of diabetes, metabolic disease, and inflammation for decades. I probably don't even need to introduce him, but
Don't say decades.
just a couple of years. So Ron will talk a little bit more about the unique collaborative nature of this study and how we've approached diabetes at the Salk.
Thank you, Reuben. Well, one of the oddities is that we've been working on these same kind of enzymes, the so-called HDACs and HDAC inhibitors, for several years, and also this metabolic switch called Ampk or AMP kinase as well. And so last year we had described something called exercise in a pill, where we had a small molecule that could confer the benefits of fitness without actually having to exercise. And it hits the same enzyme in the muscle, causing the muscle to burn more sugar and fat, and therefore activating the energy pathways in the muscle. And so as an unexpected but very important oddity is that Marc and Reuben's lab have been studying these same pathways in the liver to control sugar and glucose homeostasis as part of diabetes. And so our labs got together to really extend this platform to look at the impact of these metabolic switches on the whole body health. And this is, it's been a great collaboration, and it's one of the unique things about the Salk, which is labs that are working in allied areas have a very low energy threshold to get together and to interact and to produce top quality discoveries in a very short time. Now, the two papers really delineate or describe a new way to address a very old problem, diabetes, with a prediction of how new molecules can be used very quickly to actually cure or treat these diseases. Diabetes is one of the most vexing problems of our time, and these papers and also the support that we've received to get this work done in a relatively short time has been very impressive. So we're a small institution, but hopefully a big impact comes out of the kind of work that we do. Basic research that we're doing, which is what has really motivated these studies, has led to direct implications for therapy in people, which we think will actually move along very quickly.
Thank you, Ron. We're now able to entertain questions both from the people who are here, and we also have an internet connection. I don't know if we have anybody on the phone as well, but we're okay.
I'm Liz Malloy from the Daily Transcript. I was just wondering if you could be a little more specific about some of these cancer drugs that use this, and maybe if you know what companies are working on them. And
Well, there's a number of different companies actually that are developing drugs against these subclasses. I can't tell you them by name right off the top of my head, unfortunately, but I can quite easily get you that information. But this has been a major push in the past, really the past 3 years or so. So this is all relatively new, but there are a variety of different clinical trials. Of course, each company gives the drugs unique names, but this is a new class. Prior to this point, prior to a couple of years ago, no one had thought to try HDAC inhibitors as cancer drugs. So this is also very timely to find that they could also have something to do with diabetes, because 5 years ago they weren't being tested for any of these things.
So Merck actually has one HDAC inhibitor that is approved called SAHA, and that drug has been approved in a very small market for treating cancer, but it is approved. There is another company called Syndax that has advanced HDAC inhibitors in clinical trials, also for cancer. And I think the real important step forward here is that while HDACs are believed to be an important new direction for cancer research, the discovery here says that they may have an entirely new application towards metabolic disease, specifically diabetes. And that's a very important extension because these are molecules that are already in the clinic. And so based on this, it would be very easy to transition drugs that are in people for a new use. But no one knew to do that until this study.
But you would still have a long timeline here. But you would still have a long timeline here anyway because you've
Yes,
gotta
but you
go
don't,
back.
yeah,
Yeah.
absolutely correct. You still have a long timeline. On the other hand, you have molecules that have gone through all the drug testing, the chemistry, the toxicology, and safety studies. So we know a lot about them and what they do and what their side effects might be. So they're ready to go for this kind of test. So it accelerates it tremendously.
We have a question on the internet from Diana who asks, would HDAC inhibitors potentially work for type 1 diabetes?
Well, Yeah, that's a good question. Glucagon, the pancreatic hormone, is known to be more active in some type 1 diabetes, diabetic individuals. And so the consequence of that is that the production of glucose by the liver would be elevated. So HDAC inhibitors then would provide some relief in that setting to that group of individuals.
I'd just like to make an observation from listening to you all. I think there's been a sea change in drug development, although the pace of drug development is still long dictated by the necessity to do complex clinical trials. I think that the biomedical science now is in a position to identify specific targets rather than taking a drug like metformin, which was discovered by isolating substances from plants and seeing what happens. You can actually now find a target, in this case HDAC, looking for HDAC inhibitors, and then look very specifically for compounds that block a pathway or block an enzyme or activate a gene, and in so doing reduce the time and increase the specificity of the kind of treatments that occur. The second thing that's, I think, should be rather striking is you hear cancer and diabetes in almost the same breath by people who are working in one or the other field. And I'm just curious if anybody wants to comment on what is this relationship between cancer and diabetes in terms of your research?
Well, I think that there's two ways to look at the interaction between cancer and diabetes. One way is that we are in kind of an obesity epidemic. That is, the United States as a whole, as a country, is about 5 billion pounds overweight. That's our collective shared adipose problem.
It's getting up there with the national debt.
It's getting up with the national debt, and maybe we should put a ceiling on weight as long as we are going to do it for our budget. But you can't, and you can tell people to exercise or eat better, but that in reality doesn't work, and so you do need ways to intervene. But we know that along with increased weight and obesity is an increased risk for cancer. We also know that cancer cells undergo a profound metabolic change, and so the cancer metabolism has become a very big area. So for those of us who are studying metabolism and studying cancer, the link between these two seemingly separate areas actually at the level of the genome happens to work with several common pathways because they're both dealing with either consuming energy, which is what happens in cancer, or storing energy, which is what happens in obesity. And these are the yin-yang of a very common genetic or genomic switch that we're trying to dissect here. And so this is a very profound link. The other thing that I think I'm going to turn over to Reuben is that there's been a number of important breakthroughs in the last few years that link metabolism and cancer, and it says that this is a key area in which you need to investigate if you want to address this problem.
Yeah, I can just add a few words. My own experience from this is that I was trained as a cancer researcher, and so the major focus of my work actually originally has always been in cancer research. I was trained at MIT and then Harvard Medical School, and the discoveries that led kind of to many of the things that we're talking about today actually came from when I was there. I discovered that a tumor suppressor gene, so a gene directly mutated in human lung cancer, the third most commonly mutated lung cancer gene, is in fact the key regulator of this Ampk enzyme and is actually part of the target of that type 2 diabetes drug. So that finding very directly linked pathways that go wrong in cancer and pathways that go wrong in diabetes. And it meant that drugs that are currently used to treat diabetes, in fact, may even be useful for treating some types of cancer and vice versa. So in a sense, it's actually that connection, that nexus of cancer and diabetes that is the center of our work. And a number of other laboratories around the world in the past 5 years have made more and more connections between basic cell metabolism and cancer. Metabolism, of course, long known to be deregulating diabetes, but now both are getting more and more intertwined. And so we're really excited that here at the Salk we have such a already existing efforts in this area. So.
Good. Other questions? There's an internet question: how soon will HDAC inhibitors be available for diabetes treatment? And I think I could probably address that. I think the question is whether there is a drug that has already been tested in clinical trials for some other application, namely cancer, that might in fact be effective. If not, it would require both finding the compound, but then going through exhaustive toxicology and then clinical testing. So the pipeline is definitely years away, even most optimistically, unless there happens to be something that's already been taken fairly far along and found not to be effective for another application that might work for this.
Well, there's a potential way it could go quickly, and that is if one of the existing approved compounds does show promise in diabetes, physicians are allowed to prescribe drugs off-label. That is, it wouldn't be covered by insurance companies, but it could potentially get into people relatively quickly. But Dr. Brody is 100% correct. The trials for diabetes are actually long trials because in cancer, the trials are a little bit shorter because the nature of the disease. With diabetes, it's a chronic disease, and therefore you have to do the trials over a longer period of time with many more people. So the approval for diabetes could take several years, but the initial use of the drug in metabolic disease could actually be rather soon if one of these two pathways actually does work.
One point I just want to clarify on that, based on our work in the mouse models of diabetes, is that for any drug to be effective in this setting, it needs to inhibit the specific forms of HDACs that we found to be involved. So many of the cancer drugs might be effective against other isoforms of HDACs. That might be why they're effective in cancer. There are in total 11 different flavors, different types of HDACs in the cells. And our study has found 4 specific HDACs to be involved in this process of creating glucose. So what you really need is compounds that will target those exact forms, not all HDACs, but actually these specific ones. So it's perhaps more likely that compounds that were found not, that came out of screens for drugs for cancer, but were not perhaps effective for cancer, but will be effective against this subset of the HDACs that are involved in glucose and diabetes.
Well, obviously, you don't have a specific drug yet, but I'm curious if your research showed that a drug that would come from this, is it just a drug, or would it have to work in tandem with exercise and diet and all the other things that we currently prescribe for people with diabetes?
Well, from some of our preliminary studies, and this is not fully contained within this publication, but we think that just drugs against the HDACs alone, that can lower blood glucose somewhat. However, in practicality, you're always going to want to combine any of these treatments. Same thing with metformin. Any diabetes indication, any metabolic indication, if you can combine with exercise, the benefit is really dramatically better. So this should not be viewed as an alternative. Exercise will always help.
Yeah, let me just add to that. You know, part of the problem is that not only the liver produces too much glucose, but the muscle, which takes up a lot of the glucose and is really part of that metabolic engine, is the one that burns it. So insulin's important in allowing the glucose to go into the muscle. And so type 2 diabetics and people with insulin resistance, you know, essentially are deaf to the effects of insulin. The cells don't hear that signal, so the glucose can't go into the muscle. And so what you have is a problem of not only too much glucose being made, but too little glucose going into the tissues that need to take it up. So the likely circumstance is that these HDAC inhibitors would be used in conjunction with other insulin sensitizing drugs that are currently on the market.
Yeah, just to expand on that, what Marc said, that and it's an important point to answer your question about exercise, is that if you don't exercise, the door to glucose or sugar is closed. And if you do exercise, that door opens up and it lowers your blood sugar. You don't have to exercise a lot. Even mild exercise is as good as any drug. The problem is that we know that the American society basically is under-exercised. We do not get enough exercise as would really need for our health. And that's why these drugs become very important. In the context of insufficient exercise, they can really help quite a bit.
So would these be drugs that you would take for the rest of your life, or are you expecting some type of conversion where, you know, hopefully you could reverse the diabetes, or what do you see in these drugs down the road?
Well, you know, you can't. That's a difficult question. You know, if the diabetes is due to obesity, you can take the drugs to jumpstart a very effective treatment, and in doing that, begin to lose weight and do some exercise to get your whole body into a more healthful state. So in principle, you could get off the entire thing if you could begin to resolve the initial problem, which is the obesity. But you know, that's not always going to be the practical solution. So it depends. That will depend on individuals and the other parameters. They're more social.
I just want to add that, you know, the problem with insulin resistance and high glucose is not just the high glucose. It's the chemical changes that occur in other cells of the body that cause complications like blindness, nerve damage, heart disease. And so these are the complications that really we're trying to avoid, because those are ultimately the ones that cost the most. And so by taking these drugs, I think those complications can be avoided the best.
So we have another internet question from Diana. How collaborative have these studies been, and was this done by design, or did they just overlap coincidentally?
Marc's lab and Ron's lab and mine are all part of the Center for Nutritional Genomics, which was established just a handful of years ago by a very generous grant from the Leona and Helmsley Charitable Trust. And what that grant has allowed us to do is to actually, first of all, create an infrastructure and buy a series of cutting-edge scientific equipment that allows us to visualize glucose, to create specialized cells and a number of specialized assays that not only help each individual lab, but we've done this all together. So this funding really nucleated the fact that we were all already working on these intersecting areas, but by receiving this funding and then establishing this new metabolic core here at Salk, that really has fueled the interconnections between the labs. Now, in practical scientific terms, in fact, Marc's lab with John was independently studying how fat was burned in the fruit fly in this particular instances. And my own lab was studying how these type 2 diabetes drugs work. This goes back a couple of years now. And I think this is an interesting story of how science is done and just how the convergence of what the important molecules are. And so independently, we had come to the discovery that these HDAC enzymes were playing a role. But because our labs work together so much all of the time, when this first came up, it's like, could this be possible, that it's actually the same core set of enzymes controlling these processes, which also had not been known before? That was more the surprising part, that something so fundamental had not been appreciated. And so from that point forward, then, the studies ran parallel, and we both worked them out and deliberately worked through all the nuances together. But this never would have been possible in many senses, many aspects of this research, if we had not actually received this funding and set up this infrastructure in core. And that also includes a lot of the work in Dr. Evans' lab, which also contributed to this study. So I suppose you could say part of it was an accidental benefit, and then the other part was completely by design. we hoped funding like this would allow for these types of opportunities.
I think there's no question that serendipity plays an enormously important role in scientific discovery. At the same time, there's a wonderful quote by Louis Pasteur who said, "Chance favors the prepared mind." And so I think it's a combination of diligence and hard work and focus, but also bumping into somebody who has a discovery that might seemingly be unrelated and figuring out that, in fact, they are related. So I might ask Professor Thomas, I get asked this all the time, well, Bill, you know, you're working in fruit flies or mice. What does that have to do with anything?
Yeah, I feel like the poor cousin invited to dinner here at the table. Yeah, so you can't really get any more basic than studying a fruit fly. I mean, it's just a tiny little organism, but it turns out that in terms of genes, essentially every gene in the fruit fly has a counterpart in human. And it's estimated between 75 and 80% of human disease genes have counterparts in flies. In fact, a lot of the pathways in human diseases, such as cancer, now diabetes, are also present in flies, and they act in the same way. And so what we can do is take advantage, and this is what Marc and I have done over the past 3 or 4 years now in various projects where we've collaborated, this being just one of them. We've really taken advantage of the advanced molecular genetic tools available in flies. So we can take genes, we can express them in any cell that we want. In cells that normally don't express them, we can knock them out in any cell that we want very, very quickly. As Marc alluded to, it's got a very, very rapid generation time. And so by using the fly, using Drosophila, the fruit fly, we can rapidly go through and actually identify pathways and genes and then go into the mouse model, which Marc does, Reuben and Ron, and actually take a look into the mammalian system whether things are happening in the same manner. And it turns out in this case they are. The fruit fly is just a little mouse, a little human. It's not always the case, but the majority, the majority, as I said, I mean, the majority of genes are shared between the two organisms, and in many cases do act in the same pathways, and when you mutate them, give rise to similar conditions, diseases in humans. You could think of diseases in flies, we call them mutations.
And I think, as was pointed out, because they reproduce very quickly,
Yes.
and they don't need a big diet to feed them, it's both relatively cost-effective and very time-effective. to use
Yes,
these models.
so these mutations that don't store fat, I mean, flies in the wild feed sort of serendipitously. I mean, they'll feed, then there'll be a period of fasting, then they'll feed again. And a fly that's lacking this mutated genes in this pathway starve to death. And they starve very quickly when they're deprived of food because they're not storing fat. And so that's the phenotype that we see, and then it's translated into the mammalian system, and we get what is described here.